Test, test. Test, test. Test, test. This is a terminal. Test, test. This is Jupyter Notebooks. Test, test. This is IGV. Test, test. Again Jupyter Notebooks. Test, test. Aardslappe resources. Test, test. SCENIC Plus notebook. With target results. Cytoscape. Hello everyone. Thank you for joining this webinar on SCENIC Plus. Today we'll try to quickly walk through the whole SCENIC Plus workflow from installation until a finished output. If you have questions during the webinar, you can always type them in the comments. We'll monitor them actively, and at the end of the presentation, we'll answer the questions. Also, the questions you already sent via the sign-in page are already incorporated in the slides, so I will answer them while I go through the slides. So thank you everyone for joining. We're starting the webinar right now. So what is SCENIC Plus? SCENIC Plus is a tool to infer enhancer-driven gene regulatory networks. And for this, we make use of single-cell multiome data. And SCENIC Plus consists out of three main steps. The first step is to analyze the single-cell ataxic data. And for this, we make use of PySys topic. Then the next step is to find co-accessible regions in this single-cell ataxic data, which we analyze using a tool called PySys target to find motifs enriched in these regions and to link transcription factors to potential target regions. And finally, we incorporate the analyzed single-cell ataxic data and the motif enrichment data with single-cell RNA sequencing data to generate the actual GRNs. And for this, we have three tools on GitHub. This is PySys topic, PySys target, and SCENIC Plus. And in the newest version of SCENIC Plus, the PySys target and SCENIC Plus steps are combined into a snake-make pipeline. And this we will also show today how to use and how to run. There's also some steps in SCENIC Plus that are less easy for non-model organisms. So these are organisms that are basically different from mouse, fly, human, or chicken. And when this is the case, we'll indicate this with this hexagonal logo. So like I said, there are three main repositories. So this PySys topic, PySys target, and SCENIC Plus. You can find them on GitHub. Also, if you have issues, please post your issue on the respective GitHub page. For PySys target and SCENIC Plus, I strongly recommend to use the development branches of the code. And this is also the workflow we will be showing today. So for PySys target and SCENIC Plus, we'll be using the development version, which is a lot faster and more user friendly. Okay, so I'll quickly show how to install SCENIC Plus. This should be easy. So the first step is to clone the GitHub repository. Then I also strongly recommend to install SCENIC Plus into a new environment using Python version 3.11. So I'll make this environment using Mamba. You also can use Conda or a different package manager. And I specify that I want to use version 3.11. This step can take some time. So while SCENIC Plus is installing, in a moment I'll also start going through the slides a bit. So yes, I want to install Python. And then we activate the environment. And I'll cd in the cloned GitHub repository. Now we're still in the main branch of the code. So like I said, for SCENIC Plus, we're using the development branch. So let's check out development, and then we can simply do pip install this code, and this will install both PySys target, PySys topic, and also SCENIC Plus. So while this is installing, I'll come back to this later. I'll go over the slides a bit. So like I said, there are three main steps in SCENIC Plus. This is the PySys topic, which is used for analyzing single-cell ATAC-seq data. It can also be used as a standalone tool to analyze single-cell ATAC-seq data outside of SCENIC Plus. And the same can be said about PySys target. So this can be used as a general tool to analyze PET files, basically to look for enriched motifs. We'll start today with PySys topic. So this is the tool to analyze a single cell ATAC-seq data. So that is its purpose. And also it will impute dropout. So that's an inherent feature of single cell ATAC-seq data, that it's very sparse. Certainly if you compare it to single cell RNA sequencing data. And for that reason, using the topic modeling, we can also impute chromatin accessibility, and this will be important for running SCENIC Plus. And then finally, it will also find co-accessible genomic regions, which we can analyze using PySys target later on. The input into PySys topic is a fragment file, and one per sample, together with the index of that fragment file, and also a cell type annotation. So for each cell barcode, we need an annotation, a cell type annotation. There's also a way of running PySys topic without cell type annotation, but this I won't show today. And if you want to do this and you don't know how to do this, please have a look into the GitHub issues, because this question has already been answered. And if you still have questions about this, you can open a new issue. The output of SCENIC Plus will be a set of consensus peaks. This is a bed file, the imputed accessibility matrix, and then sets of co-accessible regions. So these are topics and differentially accessible regions. Okay, so in the meantime, I think SCENIC Plus should be installed. Okay, it's still installing, it's almost installed. So let's go over the slides a bit more. So the main workflow of PySys topic looks like this. So we start with our fragment file and cell type annotation. And we use the cell type annotation to call pseudo bulk ATAC-seq profiles. So this basically means that we aggregate all of the reads coming from cell barcodes of the same cell type into a single fragment file, and this is what we call a pseudo bulk peak set. And then we use these pseudo bulk fragment files to call consensus peaks. So we first call peaks on each individual pseudo bulk fragment file, and then we merge all of these peaks per cell type into a single consensus peak set. And then there are also some QC steps where we filter high quality cell barcodes. And once we have done this, we generate a count matrix on which we perform topic modeling to get topics which are sets of co-accessible regions. And we also use this topic modeling to impute accessibility. And while I was explaining this, SCENIC Plus is really installed, And to check your SCENIC Plus installation, you can just run SCENIC Plus in the terminal, and this should show this output with a SCENIC Plus logo. You can do the same for PySys target. So it should show the PySys target logo, which is a bit messed up. So I'll resize the terminal like this, so you can see this nice logo, and the same for PySys topic. So if you can run all of these commands and you see some usage message, this means that everything is installed correctly. Okay, now we'll quickly run through basic PySys topic analysis. And in this webinar, we'll be using data coming from mouse brain, and this is single cell multiomics data. And we'll start with the single cell ATAC-seq side of the data on which we'll run PySys topic. Okay, so the first step of the analysis is to, of course, import PySys topic and see that everything is running. So like you see, we have PySys topic version 2. Then we set some basic output directories, working directory, data directory. You can change these settings. It's just to make your life a bit more easy. Then I'll also show an overview of different samples we have. So we have fragment files, which are multiple samples, as is listed over here. So multiple donors. And for each fragment file, we have an index, as you can see, based on the TBI file. We also have already cell type annotation for each of these files. So this is something you have to do outside of PySys topic. If you have multiome data, you can start by analyzing the single cell RNA-seq side of the data using tools like ScanPy, where you perform clustering on the data and then call cell types. This is also what is done over here, but this is outside of the SCENIC Plus space, as you can say. So this is done already. So we have an N data with some cell metadata. And as you can see here, this the index of this data frame happens to be formatted a bit weirdly. So you have the sample ID at the start, then the cell barcode. For PySys topic, what we want is a column in the metadata data frame with just the cell barcodes. So I'll use this piece of code to strip out this first sample ID to only retain the cell barcodes. And then we have in our metadata data frame a column with the barcodes, and then a second column indicating the sample. The reason why this sample ID is appended to the cell barcodes is, of course, to avoid that there are some barcode collisions. But because we retain here both the information about the sample and the barcode, it's fine to have this barcode column here. And these cell barcodes should match the cell barcodes in your fragment file, as you can see over here. Otherwise, you will have errors later on. Then once we have our data, so we have our fragment files, we have our cell type annotation, the first step is to call pseudobulk ATAC-seq profiles. And this basically means that we will aggregate all of the reads coming from cell barcodes of the same cell type into a single file. And we'll use this annotation based on the Bakken et al. Paper to generate these pseudobulk ATAC-seq profiles. So as you can see on the right, you have multiple cell types, and each cell barcode is annotated to a single cell type in this case. Okay. Then to call this pseudobugs, we need to download crom sizes from UCSC, or you can provide them locally as well. You don't have to download them every time you run the code, of course. And we also have to define a dictionary where we map our fragment, our sample IDs, to the location of our fragment files. So I'll show this dictionary. So it looks like this. So we have the different sample IDs and each map to a single fragment file. And this will then go into this function called export pseudobulk. And what this function will do is, like the function name says, it will generate the pseudobulk ataxic profiles. We'll also give the function our cell-based metadata. We also give based on which cell type annotation we want to call the pseudobulks, and we also have to give the sample ID. And then once we run this function, it will generate these pseudobulk ataxic profiles. I won't run this function live because it will take some time to run, but I've already run it before. And once we have these ataxic pseudobulk profiles, we can also visualize them in tools like IGV or the UCSC genome browser. And this is what I'm showing over here. So here you can see for each of the cell types the ataxic profiles, the pseudobulks that we called. And this is in the SOX9 locus, as you can see here on the bottom. And you can see that for astrocytes for which SOX9 is a marker gene, that we have a clear peak over here, which is less present in the other cell types. Okay, so now we can continue with our workflow. And the next step is to infer consensus peaks. So what this step will do is it will, here we will call peaks for each of the cell types. So for each of the cell types we see here in the IGV window, peaks will be called. And then these peaks will be merged into a consensus peak set. So this step requires actually two steps, so two functions. And the first step is this peak calling function. So this will do the peak calling per individual pseudobulk profile. And then later on, so this will create, this uses max2, and this will create a narrow peak file per cell type. And then we'll use the get consensus peaks function to merge all of these peaks into a single consensus set. At this step, also each peak will be resized to the same size, which is controlled using this parameter, peak half width, and is set to 250. So this means that the peaks will get resized to 500 base pairs. So this is the half of the actual peak size. After running this function, you can, again, I won't run it here, you can export these peaks to a bed file, which you can also visualize in IGV. So if you go back to the IGV window at the bottom, you can see in black the consensus peaks that have been called. And as you can see, yeah, there are quite a lot of consensus peaks spanning yeah, a large part of the genome, you could say. And we'll use these peaks as features to create a count matrix. So what we'll do next is for each cell barcode, so not for each cell type, we'll create a count matrix with the peaks as features and the cell barcodes as index, and then we'll count the amount of reads per consensus peak. But before we can do this, we'll also do some QC. And the reason for this is that not all cell barcodes in the fragment file are of high quality. So they are not all high quality, don't represent high quality cells, or they don't represent cells at all. So they can be noise. And for this, we have some QC steps, and this is also new in the development version of PySys topic, where we have a command line version to run the QC. This step is now a lot faster as well and more efficient to run. So I'll quickly run through the QC. So the first thing we need to do is we need to get TSS annotation. The reason for this is later on we'll calculate TSS enrichment per cell barcode, and for this we of course need to get the TSS of each gene. So we first run this first command to get the correct annotation from ensemble representing the gene names, and then using this annotation that we get over here, we can get the actual TSS file. So this will be a BET file that will be downloaded from ensemble. And then next, there's a PySys topic QC command that takes a fragment file, so one per sample, your consensus peaks, the TSS file that we created before, and output the quality control metrics. Using this short script, you can generate this command for each sample, and then you can also run this in parallel across samples using the GNU parallel command as shown over here. It is also everything I'm showing is also available in the read the docs. So if I'm going too fast, you can always go back. Also, this webinar is recorded, so you can always play it again. Okay, so this will calculate the actual QC metrics. Now we can also generate some plots. We can generate plots on a sample level. I might have just accidentally deleted my QC folder, so you'll have to believe me that we can generate these kind of plots. They are also present in the tutorial. So what this will show, I can actually maybe quickly show you using the actual tutorial. Yes, over here. So again, I won't recalculate the QC right now because it will take some time, but I'll show you on the tutorial. There we can also see the QC stats. So over here, bear with me for a second. Okay. So this is of course the risk of a live demo, but going to the QC. So again, these are exactly the same steps as I was showing. We'll get these types of plots for each of the samples. So we'll get this barcode rank plot where the number of unique fragments per cell barcode are shown. And what we want to see is this sharp knee in the plot. Then we also have the fragment size distribution, which shows the actual size distribution of each fragment, and this should show this nice profile which represents mononucleosomal and dinucleosomal fragments, and then also a nice TSS enrichment. So this is how a nice high quality sample looks like. Then we can also plot some QC stats on cell barcode level. So here each dot is a single cell barcode, and here we show the TSS enrichment versus the number of unique fragments. So this is the same TSS enrichment as shown above. Then also the fraction of reads in peaks versus the number of unique fragments, and the duplication rate versus the number of unique fragments. And we use these metrics to set thresholds to select the high quality barcode. So the barcodes on the top right will be selected, and from the new version we also set these thresholds automatically, but you can always modify them by changing these values on top. So if you set them to none, we'll use automatic thresholding. If you change the value, it will be the actual value you set. Okay, so we can go back to the actual Jupyter notebook where we had this error. Okay, and then once we have done the QC, once we have the consensus peaks, we can create our count matrix using the create systopic object function. So here we have to give the fragment file, the path to the consensus peaks, and the metrics, so the QC stats that we have just calculated. This function will run for each sample individually. And later on, we merge the different samples using the merge function. Once we have generated our systopic object, we can save it to disk. Unfortunately, still using Pickle at this time, but this will change. And then we now also have a command line version to run the topic modeling. So using the PySys topic modeling and mallet, where we run topics with different amount of topics. So we run a model with 5 topics, 10, 15, and so on. This will also take quite a long time to run. So this takes the longest time of the whole workflow. So I would suggest to submit a job for this. And then once these models have been run, you can evaluate your model using a set of methods. For this, I'll also switch to the tutorial because I accidentally just deleted these files. Okay, so back to the tutorial. To select the optimal model, we have these several metrics. So this is kind of a not super objective step, but also it doesn't matter that much the amount of topics. So what you want to do is you want to select a model with a number of topics where most of the curves start saturating. But this is of course not an exact science. There's also not a really good way to do this. But if you are a bit off, so if your model is like 5 topics less or 5 topics more than the ideal, this in practice also doesn't really matter that much. Okay. And then once we have selected the model, we'll add the model to the systopic object and save the systopic object to disk. And then we can continue with the SCENIC Plus workflow. So going back to the slides now, I can answer some of the questions you posed. So one of the questions that was asked is when you change the cell type annotations, it seems that we have to start again all the way from the beginning of the workflow. Is kind of true, because when you change your cell type annotation, yeah, you will also change the consensus peaks that you're calling. So indeed, this will affect the entire downstream workflow. So it's important to select high quality cell types to call the consensus peaks. Then someone is asking whether there are some specific features that are specific for AttackSeek and the sparse nature of this, and this is indeed the case. So the topic modeling can be used to impute dropouts, and this is also then used for dimensionality reduction and downstream in SCENIC Plus. Okay. So this was the PySys topic part. Now we can continue with the motive enrichment part. So the purpose of this step is to find enriched motifs in co-accessible regions. The input to PySys target is a set of co-accessible regions. So these can be topics or DARs. And also very importantly, a systarget database. And the output is a set of enriched motifs. And also importantly, these motifs are annotated to transcription factors. So the systarget database is an important thing here. So for this, there are multiple options. And for human, mouse, and fly, we already have pre-computed systarget databases. But these are on general regions. So for example, for human and mouse, these are on the screen regions. So these regions are generated agnostic of your analysis, and they just cover a fraction of the genome. So they're based on different, might be based on different cell types from your analysis. So we strongly suggest to create your own systarget database based on your consensus peaks. I'll also quickly show you how to do this. So the input for this is a set of consensus peaks and a motif collection. And the motif collection is listed on our website. So if you go to resources.aertslab.org/systarget, you come on our systarget website, where you can find our motif collection in the folder motif collection, and you can find the latest motif collection, which is V10 over here, which you can then also download this zip file. On this website, you will also find the pre-computed databases. So this is in database folder, and there you have a database for fly, human, and mouse. For example, for human, you can go to the latest assembly. The database is based on screen regions, or screen. And then V10, latest motif collection. We find here the region-based databases, and you always have a ranking-based database and a scores database, and we need both of them for the SCENIC Plus analysis. Then I'll quickly show you how to create these databases. For this, I quickly have to go back. For creating your systarget database, we have a separate repo called Create Systarget Databases. And this tutorial to create the database is already online on the SCENIC Plus Read the Docs website. So the first step is to, of course, clone the repository. To create the databases, we use a tool called ClusterBuster. ClusterBuster will score DNA sequences for motifs, and so we also download this tool. It uses a hidden Markov model to generate these scores. And then we also have to download the motif collection, like I explained before. Then the first step to actually run this code is to get your consensus peaks and to convert them to a FASTA file. This can be done using regular bed tools commands, bed tools get FASTA. But we also have a little script that also will generate the FASTA files, but will also add a padding of a specified length. So in this case, 1 KB. And this padding will be used as a background by ClusterBuster. You can choose to do this or not to do this. It doesn't affect the analysis too much, but I usually do it. And then we can run the create systarget motif database, where we all again specify our background padding that we used in the command before, and this will then score all of your DNA sequences for all of the motifs in our collection. Okay. Then here there were also some questions. So there was a question whether there's a way to add extra motifs to the list. This is possible to do. You just have to convert your motifs to the cluster buster format, so the CB format. And then it's also important to generate motif to, I'm not showing the slides. Then it's also important to create motif to TF annotations for these motifs, and for this you have to strictly follow the format that we also use for our motif to TF annotation. So I quickly show it on the SysTarget resources website. So you can find examples on our website under, I'm going up, under motif to TF. And then you can find here for chicken, for fly, human, and mouse, motif to TF annotations for all of the motifs in our collection. And you have to follow the format of this data frame, basically. If you have troubles with this step, because it can be quite tricky to have the correct format, you can always open a GitHub issue, and I'm happy to help. Okay. So going back to the slides. So yes, you can add extra motifs if you want. Support for other species like zebrafish. So we don't have pre-computed database for this. Neither do we have motif to TF annotations, but it's possible to also run it on zebrafish. But you have to generate the database yourself and also the motif to TF annotation. And this motif to TF annotation, what you can do is you can use the pre-existing motif to TF annotations that we have for human, for example, and then use zebrafish orthologs to generate your motif to TF annotation. Then someone was asking whether you can also use it on plant. So this we certainly have not tried, but in theory this is possible. You will need a motif collection that is suitable for plant, and then also motif to TF annotations for plant. And then the person was also asking for single-cell RNA-seq datasets. Of course, you need both single-cell RNA-seq and ATAC-seq to run SCENIC+. So in theory, it's possible if you have multi-ohm data on plant, but it will be, it will involve some custom steps that we have not tried before. Okay, and with this, we go to the last step, which is the actual GRN inference. And for this, I give the floor to Darina, who is sitting next to me.
Yes, so I will explain briefly the how we run SCENIC+. I will explain the inputs of the that we need for the different functions and how we can interpret and use the outputs for the downstream analysis. So let's get to that. Just 1 second. Okay. So first we'll need to prepare the data. So this comes from the pre-processing step that Sepp has already explained. So from the running systopic, we'll have the systopic object from which we want to binarize the topics to create sets of regions on which we will run SCENIC+. So what does it mean that we binarize the topics? We want to, for each topic, we will have regions we will have probability in each region to belong to a certain topic. And then for each topic, we want to select regions with the highest probability to belong to that topic. So we can do this either automatically using OTSU, which is a method for thresholding. And in this way, we will pick an automatic threshold based on which we select the regions that belong to these to each topic. So in this case, we had 80 topics, and from each topic, we will pick different number of regions. Another way to do this will be by taking simply the top 3,000 regions. So here we have selected 3,000, but of course you can decide on this value yourself. And then so for each topic, we take the regions which have the highest probability to belong to that topic. These we need to save into bed files, which we'll then provide to the SCENIC Plus pipeline. And it is important that this region sets folder, which will contain the bed files, has a correct structure, which I'll show in a moment. So another type of region sets that we can create will be differentially accessible regions. Oh, there's an error. Okay. These differentially accessible regions, yeah. So for each cell type, we can compute regions that are more accessible in this cell type in comparison with all the other cells in the data that we have. So we are still working on the same data set, the mouse cerebral cortex, as Sepe was showing. And from this, again, we need to compute this on the imputed accessibility rather than just the raw accessibility counts. And for that, we then compute this with a function called find differential features. Then again, we can save this into bed files within the region set folder. So I have pre-run all of this beforehand just in case, because otherwise you might delete a folder that you need for your demo. But in essence, the region sets folder should look like this. It has the name of the the sets that we want to add into our analysis, and then in each folder we will have the bed file for the set of regions that we are that we've created. So this is very important because SCENIC Plus expects the inputs to look like this. Now the next part that we will need for the inputs, or which is important to also prepare, is the AData object, so the single-cell RNA-seq data. So simply from the ScanPy tutorial, we can, if we follow that, we already do this step where we add the raw gene expression matrix into our AData object. So we need to check that this is indeed the case. So here I just double check this in the AData object that we are using in the analysis. There is indeed the raw count matrix included. Now, to run the pipeline, we do this in Bash. So I will switch to that, and should we get there maybe? Okay. So yeah, to initialize this, we'll first create a directory. Let me see where in which is this the part? Oh no, this is the folder from the okay. Yes, okay. So here I create the folder to running the pipeline, and let's call it SCENIC Plus pipeline. And here we initialize SCENIC, the snake make files. So So to do this, we just run this command SCENIC plus. And we choose the output directory as we have created. And this should normally create files in the directory, but apparently there's some problem with that. We can fix it. Okay, so now the SNAKE folder is created here. We can check it, what it looks like. And here we see the files that get created. So the most important one here is the config YAML file, where we configure the inputs and outputs of the pipeline. So what it's when it's created, it's just an empty file with some default parameters, which you can show. So it will contain the structure of the config file, where we need to provide all the input data. So the systopic object file name, the AData object, the folder for which contain the region sets, which again must have the structures I have shown before. Then we also need to provide the systargets databases that we created before, and paths to modif annotation. So I will just copy the config file as I've prepared it before into this folder. And now we can show what it when it's has all the paths filled in. So here are the database that we have for the CTX for the systarget database. This should be the regions versus motif rankings, but for this should be regions versus motif scores. And again, we also have the path to motif annotations. Now for the outputs, we can just create an outputs folder where we save all the data there. We don't need to change much, because this is just a list of all the files that get created as the output for the analysis that from the pipeline that we can further analyze. There is a temporary folder where some temporary file gets saved. And some of the parameters for data preparation that we do need to adapt is, for example, the species that we are working with. So for our case, this is the mouse, and this is also how we can download the genome annotations from Ensemble, and we want to choose the Ensemble version, which will have the highest overlap of genes with our data. So for doing this, we have a small script, which I can show you. So to pick the Biomart host, we can go to archive.ensemble.org, where we can find all the possible all the available genome annotations that are available in Biomart, and then we check how big is the overlap between the gene names in our AData object and the gene annotations in the Biomart host. So for our case, this was the largest overlap was for version 2019. So this is the one that we also input in the configuration file. And for the rest, we can keep all the parameters as they are suggested in the configuration file by default. Now, to run the SNAKEMake pipeline, we just need to. Oh, I forgot to change this. Yes. So we just need to go to the SNAKEMake directory, and here we will. We can choose a number of cores which we want to use to run the code, and let's say go with 20, and then the code starts running based on the. Okay, yeah, because I already run it. This will show. Yeah, it's all the output files already saved where they are, but normally it would show a list of jobs that would be run, and it will give you all the outputs. This also takes quite some time, so of course we cannot show that in this demo, but I will show the outputs that we get from this. So those should be in. So there are some 18 files that get created, from which we can use for some downstream analysis and to explore the results of the SCENIC Plus pipeline. So one of the interesting outputs is this CTX result. So this is the results of the enrichment so the motif enrichment analysis, which is run within the pipeline as well. So in essence, you don't need to run it beforehand. So I can show what this HTML file looks like. Okay, so this is a very long file which has for each region set, it will show the most enriched motifs present in these regions. It also has some scores, so the normalized enrichment score. So this is how enriched this motif is within these regions, and what transcription factor annotations there are for this motif. So we can, of course, have a look at this for each topic or for each region set that we have provided, and see what transcription factors would be regulating or binding to the regions within these region sets. And now going back to the notebook. No. It is good. Just getting a bit confused with this. Okay, this one. So now that we have run the pipeline, we can look at the results that we get. So from also other motifs enriched. So here I select a few examples from the DARs, because those are more obvious to look at, since they have the cell type annotations, and we can have a look at some known transcription factors. So we can, for example, for layer 6 CT, we find TBR, which is a marker of layer 6 CT neurons. Now, from the other outputs, the main output that we want to look at will be the mdata object. So it looks like this. It contains some metadata for all the regulons that we find, and it also has the computed different modalities. Well, firstly, this is simply the scRNA that we have provided, and then we have attack counts that we have also provided via the systopic object. But then we also have the AUC scores that are computed for both gene, based on the gene expression and region accessibility from their corresponding A data and systopic object. And we have this for both direct and extended regulons, where the direct regulons are based on the direct annotations of the transcription factor motifs, and the extended are based on the orthology annotation. So here we have a result of 180 direct regulons and 302 extended regions. Now, these regions are summarized in their metadata pandas data frame, where we have some information on the region to gene and transcription factor to gene relationships. So some of these, what it contains is then the region name, then the target gene to which this region corresponds. There is an importance scores between the region and gene, the correlation between the region's accessibility and the gene's expression. And then, since sometimes this can also be negative, we also store the absolute value of this. There is also the transcription factor that is regulating this, or that binds to the region. And for each region, there is the signature, which contains the name of the transcription factor, whether it's direct or not, and then this relationship. So for the first symbol corresponds to the transcription factor to gene relationship. So if it's positive, it means that they are positively correlated. If it's negative, the transcription factor expression and the target gene expression is negative. While the second symbol corresponds to the this region to gene relationship. So if it's positive, the region's accessibility is positively correlated with the gene's expression, and negative is the other way around. So yeah, that's some of the metadata that we can find for each of these relationships. And then we have also this triplet rank, which summarizes this score for all the regions, not for the regions, but for the for these combinations. So it's better explained with an image. So the triplet scores aggregates the ranking of the transcription factor to region scores, the transcription factor to gene scores, and then region to gene scores, and then it can be used to prioritize enhancers. So the transcription factor to genes regions score is based from the motif enrichment, and the other two scores we can find in the in this metadata data frame. If you don't want to load the mu data object because maybe it's quite large, we can also directly look at this metadata in the outputted TSV file, which is also within the outputs folder. And now for some downstream analysis to explore the resulting regions that we get from SCENIC Plus, we can have a look at this gene-based AUC scores that are computed for each cell for each region. So we have the direct ones and extended. It's stored within the mu data object as an A data object, and we can concatenate these into one A data object, adds our metadata to it, and the resulting A data object will then contain all of the gene-based AUC scores for all the regions, and then the metadata for cell type annotation. We can use then SCEMPI's neighbors and UMAP function to compute UMAP based on this AUC scores. And when we plot it, we can see that the cell types are clustering as you would expect. So we have nice cell type specific clusters based on the E-region scores. We can also compute specificity scores for each of the regions. So for this, we have the region specificity score function, and when we plot it, so here in the variable we choose for which cell type annotation we compute these scores, and then when we plot it, we can see for So in the plot, we can see that we will see the scores that has been computed for each regulon for each cell type. So here's for example for astrocytes, and the top. Then they are ranked by from the highest RSS score to the lowest, and the outliers here would be the ones that are the most specific for these cell types. We can also plot these enrichment scores. So if you just use the eRegulon gene AUC data object that we have created, and we will color the UMAPs by the AUC scores from the most specific regulons. So we can see, for example, for this, I think this was layer 2-3, the most specific regulon would be expressed there. So we can see that it's more expressed here than in the other cell types. A different way to visualize this is with a heatmap.plot. So for this, again, there is a function called heatmap.plot. Speaks for itself. Where the input will be the mdata object, which we can find in the output folder from SCENIC Plus, from the SCENIC Make pipeline, and then we can choose how we color the heat map. So here we color it based on the gene-based AUC, and the size. So the dot size will be the region-based AUC. We group the cells again by the same cell type annotations as before, and we provide the metadata, the features. But so this is all also in the tutorial on the website. So, but this is what we need to provide to get this plot where we can see the these scores more summarized for all of the regulons in our data sets that we have found. Well, this is only for the direct ones, but we could also do this for the extended ones. It just would be quite a lot to see it in this heat map. Now, one more thing that, well, we can do still a lot of other analysis with these results, but to visualize the networks, we can also use Cytoscape. So for this, I sorted the regulons based on their triplet ranks, taken the top 200 of these 200 rows, and then save them into a TSV file, which we can then load to Cytoscape. So I can briefly show how we can then build the network in Cytoscape. So here we just need to import the network from file, and the file should be here. We cannot really see the. Oh, yeah, okay. Well, so we will get the prompt where we can choose in the table that the transcription factor is used as the source node, and gene will be used as the target node, and we accept this, and this will lead to the network that I have also shown. So yeah, then we can do this with different combinations or different selection of the target regulons, or we could, for example, take the most enriched or most specific based on the RSS scores for a certain cell type and investigate which interactions we can find there as an example. Yeah, so I give word back to Sepa.
Okay, thank you, Darina, for this nice overview. So yeah, that was the full SCENIC Plus workflow. So for the remainder of the time, I will just be answering questions. So if you are in a hurry to leave, or you don't think it's interesting anymore, yeah, just feel free to leave. If you still have questions, you can still post them in the chat, and I'll try to answer them. So before I'll answer the question in the chat, I'll answer the questions that were posted on using the sign up link. So the first question was, when will SCENIC Plus use Yuan as its main data structure? So yeah, it's already using data at this moment in the development branch. So in the development branch, we completely switched away from the SCENIC Plus object class, and now we use data as the main data structure. We're still updating some downstream functions, but all of the functions that were shown today we can already use, of course. Then next question was, can SCENIC Plus be used to filter regulons previously called using PyScenic? Yeah, we don't really have functions for this. And then the next question is whether a single cell ataxic data is needed. It's really needed to run SCENIC Plus, so you cannot run SCENIC Plus without single cell ataxic data. You can run it using non-multiome data. So if you have separate single cell RNA-seq and single cell ataxic data, as long as you are able to match the cell types in the single cell RNA-seq data with the single cell ataxic data cell types, so based on the same cell type annotation, for example. Then how to integrate SNAP-ATAC2 and SCENIC Plus without single cell RNA-seq data. So it's possible to first start with SNAP-ATAC, for example, to already generate your count matrix, or to do a first round of dimensionality reduction and clustering. And then you can use those clusters to to generate pseudobulk profiles like I showed before. So you can use that type of cell type annotation as well. Again, you need single cell RNA-seq to run the whole SCENIC Plus workflow, but if you're only interested in the PyCystopic part, it's fine to start with SNAP-ATAC to define your clusters and use these clusters as input for the pseudobulk p-calling. Then there's a bunch of questions on how to do comparisons between case and controls, or different samples and so on, mutant cell types, for example. This question is very strongly dependent on your dataset and the research question you are asking. We also don't have functions to directly do these types of comparisons. So there's no one function you can run, but I can give some general recommendations. So the first recommendation I would give is to try and not subset your data too much. So for example, don't subset to a single cell type. And the reason for this is that SCENIC Plus uses the background of the other cell types to be able to infer the GRN. So you kind of need the other cell types to figure out what is specific to the cell type that you're interested in. What you could consider doing is to subset your data only for the cell types of interest, and then keep case and control. And as long as there is enough difference between case and control, you might be able to find some regulons that are different between the two. But yeah, like I said, there's no one way of doing this. What you can also do is you can also run it on your entire data set, and then afterwards, once you have the GRN, using the AC values, for example, the enrichment scores, try to figure out whether there are some regulons that are specific for a mutant cell type versus your normal cell type. So I would always try to use as much data as possible, and try to not subset your data if it's possible, and then do the comparison afterwards. Whether we have any guidelines on how asymmetries in number of cells per cell type affect the resulting EGRN? Yeah, this is kind of a good question. We don't really have guidelines on this, and we also didn't really test this before, but of course you need enough cells per cell type for example, you need enough cells per cell type to call confident pseudo-bulk peaks, but we didn't really assess the actual number, so a limit, I cannot say what the smallest amount is. Yeah. So are there any assumptions or pre-processing steps that are important for running SCENIC Plus? For this, I want to highlight again that's very important to check your motive enrichment results, like Darina was also showing before, because if you don't have nice motive enrichment results, you also won't have nice networks. So everything starts basically from the motive enrichment results. Is if those are not nice, if you don't see nice motives coming up, then the SCENIC Plus output will also not be good. So really check your motive enrichment results. That's the most important. Whether there are any key parameters to change when using spatial RNA combined with ATAC-seq. This we also didn't test before, so I cannot really answer this question. So you'll just have to try it out. But I cannot think of any particular parameter that you have to change. The only thing is that you have to be able to match the RNA-seq cells with ATAC-seq once. Then a bunch of questions on scalability. So like I was showing now in the tutorial, we were showing the development branch. This is a lot more scalable, so it's using a lot less memory. So I would really suggest to use the development version of SCENIC Plus, which will, by the way, become the default version quite soon. So you better already get used to it if you are starting your analysis now. Whether you can run it without internet access on the cluster, also a good question. So yeah, indeed, some steps are showing that you have to download files from UCSC, from Ensemble, and so on. For this, you need internet access, but you can also provide these files yourself. So download them beforehand and then upload them to the cluster, and then you can still run the pipeline. But yeah, you need some modifications from the tutorial, but it's perfectly doable. Does the result get different each time you run the pipeline? Yes, it's possible that results differ. There are some stochastic steps, but in general, I would not recommend to run the pipeline multiple times. From what we see, results are quite robust, but there are some small differences that arise, certainly if you change the seed. How to select the best irregulons. Indeed, we have multiple metrics for this. So we can, for example, calculate the correlation between the region-based and the gene-based AUC values, or the correlation between the TF expression and the AUC values. And then we also have the triplet ranking to select irregulons. Again, there's no, and then also the RSS score. So there are there are multiple ways to select irregulons. What is the best method? I don't really know. I kind of like the triplet ranking, and also the TF expression to AUC based correlations are often quite good for finding known transcription factors. So if you're selecting a method, I would always first try to focus on a cell type where you already expect to find some transcription factors, and then see whether you can recover the known transcription factors, and then you can go to your new cell types. Okay, so these were the questions that were submitted in the form. Then there are also some questions from the chat, which are on my other screen here, so you cannot see them, but I'll read them out loud. So one question is whether the development version is a different but just more efficient version of the original release, and whether you get the same results if you run development versus non-development version. So indeed, it's supposed to be the same pipeline. The functions that are used are also have the same purpose. So the results should be similar, but there might be some, I cannot guarantee that it's 100% the same. So, but in general, the results should be similar enough. Is a memory problem partially alleviated for the development branch? Yes. Development branch is a lot more efficient, so use that branch. Whether we plan to provide any instructions to create a singularity image for the development version? Yes, this will also come on GitHub online soon, how to do this. I didn't get around to it yet. Then there was a question how to generate the cell type annotation data frame. So this most of the time, if you have multiome data, this comes from the single cell RNA-seq analysis, where you can annotate the cell types based on known marker genes. If you don't have single cell RNA sequencing data, this annotation comes from something else. For example, you have already run a snap attack, like some people are commenting in the on YouTube, and there you have cluster labels for each cell, so you can use those cluster labels. Or you can also run systopic first using the screen regions and then perform topic modeling, and then based on those topic models generate your clusters, and then use those clusters to generate cell bugs. That's also possible. Then there was also a person, or the same person was asking whether it's an issue if you do QC before generating these labels. So meaning that there might be some cell barcodes for which you don't have any cell type label. This is no issue at all. So like I was showing in the tutorial, there also for some cell barcodes we didn't have a label. So these are the cell barcodes that we filtered out in the RNA-seq part. This step is only used to generate the cell type annotation is only used to generate the pseudobulk profiles. So it's fine to only use a subset of barcodes for this step. So later on in the QC is where the actual barcode selection will occur for the single cell ATAC-seq side. But for cell type annotation, it's not needed to have annotation for each cell barcode. And then again, the same person was asking, yeah, with the snap attack, you get, instead of these regions, you get tiles across the whole genome to create your count matrix. This is indeed very inefficient for bicystopic, because yeah, you'll have a huge count matrix. So in this case, I would recommend to use the cell type annotation from snap attack to create again the consensus peaks. Okay. Then there was a question whether in general you would select a higher number of topics than cell types. In general, this is the case. So I would say at least the number of topics should at least be the number of cell types, but often it's more like double the amount. Is a good estimate. Then some person is commenting that using pickle or dill to store data is very memory inefficient. Yeah, this is true. This is also why we are moving away from pickle and dill. So for SCENIC Plus, there's no pickle anymore. For PySys Target, also no pickle. Only for PySys Topic. We still have some things we save using pickle, but here we're also moving away from this and moving to NData. Then there are some metrics for topic selection that are supposed to go down as the so these metrics go down as the model performance increases. This is true, but in the plots for those metrics where the number gets smaller as the performance of the model increases, we plot the metric inverted. So this means that in the plot, the numbers will go up. So if you look at the plot of the topic selection, all numbers should be going up. But in reality, some of the metrics, for example, iron, will be going down. But in the plot, it goes up. If you only have RNA but no attack, would you recommend to use PyScenic or SCENIC? So PyScenic is a Python version, SCENIC is a R version. I would strongly recommend to use PyScenic because it's a lot faster and also more maintained compared to SCENIC. And indeed, attack is mandatory for SCENIC Plus. Whether we'll share these notebooks, yes. So the notebooks I shared in the seminar will be shared as well if you want. I'll put them on with the docs. And also the recording will be shared. Then there's also a person asking whether there can be a group chat to talk about SCENIC Plus. There actually already is kind of a group chat. You can, so we try to use the SCENIC, the discussion tab in the GitHub page to talk about results. If you have general questions, you can use the discussion tab. I think this is quite nice. Feature of GitHub. So I would use the discussion tab. What's the benefit of using SnakeMake over raw Python? So you can also run the SCENIC Plus code using just command line scripts. The benefit is, first you don't have to run it manually, so you don't have to babysit your code. That's one benefit. Also, SnakeMake will optimize the different steps. So when it notices that a certain function doesn't need all of the cores, it will launch another command so that two steps can run in parallel, as long as they don't depend on the same input, of course, if they can run independently. So that's the benefit. Can you obtain cell type specific EGRNs from SCENIC Plus? Yeah, this is difficult to get. So this is a downside of SCENIC Plus. You get one network across all of your cell types, but the network is not cell type specific. So the edges are across cell types, not within a cell type. There's no easy way, unfortunately, to do this. Then I also want to make a note on Biomart. The selection of the server is not only important for the overlap with your G names, but also for the assembly. For example, if you're using MM10 data, now the default version of Ensemble is MM11. So you have to go to Biomart archives to select the correct archive version. I can maybe quickly show you how to select this. I have a website here. So if you go to, I'll just make this a bit. I can find my window. Yeah, over here. So so if you Google Ensemble Archive, you'll go on this Ensemble Archives website, and then you can look here at the table, and here you have all different species. For example, if you search for mouse, then you find here that the latest version is M39, which is MM11. So if you are using MM10 data, you have to go back to a previous version of the archive. So you can select, for example, this one. And then you just copy the link which is shown on top of the web page to run your analysis. So that's also important. Okay. Let's see if there are more questions. I'm reading right now. Whether RSS is some kind of statistical test for regulons that are more active in a subset of cells. Yeah, so this is a metric of entropy. So it's an entropy based method to calculate a specificity score, but it's not a statistical test in the sense that there is a p-value, or I can also not say this cutoff you have to use on the RSS score to select your Regulon. So it's not the best score for that. Someone asking why, if you generate UMAP using PySys and ScanPy, yeah, I guess both on single cell ataxic data, why the plots are different. I would submit a GitHub discussion for this where you can show the code you're actually using to generate the plot. The code that Systopic and ScanPy uses to generate UMAP is also slightly different. But if they are very different, yeah, I would use an open discussion. Whether you can use pre-existing analysis from SIGNAC and Cromvar with SCENIC Plus. Yeah, in theory this is possible, but of course we don't really have tutorials for this. We cannot cover all of the tools that are available, but as long as this will be quite hard, of course. You have to fit to the data structure that SCENIC Plus is using, but if you are able to do this, you in theory can do this, but we don't have any functions to specifically do this. Then someone says whether my recommendation about not running SCENIC Plus on each cell type separately also applies to SCENIC in general. Yes, I would also say it applies to SCENIC in general. Yeah, someone asked whether to make a cell type specific each run, you can do this afterwards. Indeed, that's also how I would do it. So based on the expression of TFs and target genes, you can try to make your network more cell type specific. There's also someone asking whether there will be a Discord channel for SCENIC Plus. There actually also is a Discord for SCENIC Plus, but no one is using it. But I can post this on GitHub if people like to use Discord. It exists. Yeah. Yep. And then someone is asking that PyScenic has Nextflow. Why did you switch to SnakeMake for SCENIC Plus? The reason for this is because we also have a collaborative project with other labs that are also using GRN inference methods, and they use SnakeMake, so to be more in line with them. But in theory, also Nextflow would be fine. Is it possible to access the final data used in the dot-plot heat map? Yes, this is possible. If you want to get the actual code, I would to do this. So if you want an example, please post this question again in discussion, and I will send you an example how to do this. Yeah, okay, that's the last question I have here in my Google Docs. Okay, and also the last question. So if there are no more questions, we can end the webinar here. I hope you found it kind of interesting, and have a good end of your day if you're close to Europe, and otherwise a nice day if you're not close to Europe. Okay, see you.